I'm Dr. Mark Gold, and this is the Gold Standard. Chuck, thanks for joining me for the Gold Standard.
Mark, it's a great pleasure to be here with you today.
Well, you are the gold standard in many ways, and that's a good lead into thinking about Washington University in St. Louis. I mean, I was an undergraduate at Washington University in St. Louis and met John Feighner before I graduated and toured the Department of Psychiatry. At one time, I was a history major, and another time I was an English major, and then I was a psychology major when I met John. And at that time, he was working on the DSM. And I don't know if people remember, but WashU really invented organized psychiatric diagnosis. Maybe you could just summarize that Feighner and Robbins, Guze, the kind of original work on diagnosis and how that's progressed over time.
Yeah, sure, Mark. And it's one of the hallmarks of this department here at WashU. The home of diagnostic criteria and medical model psychiatry in many ways. And the story of this department kind of grew out of the 1950s. It's been around longer than that, but really the 1950s is when things really started to change here at WashU. So Ed Gilday was then the head of the department, and he recruited three amazing young psychiatrists. So Sam Guze joined the department, Eli Robbins joined the department around that time, and so did George Winokur. And they were given a full range of trying to make decisions about how to move the field of psychiatry forward. And they believed very strongly that psychiatric illnesses had to be studied as medical illnesses. And that meant you had to have very reliable ways to make the diagnoses. So they set out about codifying some criteria for making a number of diagnoses that they thought were validated. Validated means there's evidence that across standards that these are probably illnesses that will run true to time. And that what they were doing is a lot of clinical research. And by the time you get to the late 60s to the early 70s, which is around the time John Feighner was here, they were using criteria for diagnosis and research, but had never codified kind of what they were doing. So it was really Feighner who actually pushed them to do this. And there's an incredibly famous paper from this department that John Feighner was the first author on. It was in the old Archives of General Psychiatry, and it was about diagnostic criteria for psychiatric illnesses. Feighner was actually the chief resident at the time when that paper. Is that amazing, Mark, or what? I mean,
well, that's how. I was the first author on the clonidine invention paper as a chief, and I think that paper was probably 71 or 72, and it would be about the time I met him.
Yeah, that's exactly right. So that original paper had 14 criteria for 14 what they believed were well-validated psychiatric illnesses. There were actually 15 diagnoses in the paper. They considered depression secondary to another psychiatric illness or medical illnesses to be a separate diagnosis. So there were 15 diagnoses in there, much fewer than we have today in the DSM. Well, what ends up happening with the Feighner criteria is they start to catch on by the mid-70s or so. And working with Robert Spitzer, who was at Columbia, they came up with further criteria for research and was called the research diagnostic criteria. So that was a Columbia and WashU kind of collaboration kind of on that. And then that became the basis that led to the major change in psychiatry, which was DSM-III, published roughly 1980. That was the first time the psychiatric DSM, Diagnostic and Statistical Manual, actually used criteria for making psychiatric diagnosis. Before that, it was clinical impression and clinical expertise that led to the diagnosis. So DSM-III was really the transforming moment for broader psychiatry. Well, that caught on and became the diagnostic standard worldwide, and everything that flows from it since that time traces its roots back to DSM-III and back to the Feighner criterion in effect.
It's really amazing and critically important because, of course, if you don't have diagnoses, how could you have a clinical trial? And if diagnoses just depend on countertransference, you're pretty limited.
Yeah, and that was the order of the day at the time. So psychodynamics kind of ruled the field of psychiatry, which, by the way, was never the case here at WashU. From the 50s on, that was never the case. So they were really pretty free to kind of go just pursue their own ideas in that. National Institute of Health funding wasn't a big driver behind the research. It was their clinical practice, their teaching, and the clinical research that they did as a function of that were the drivers behind how they created the criteria-based diagnoses.
Did you ever work with these guys?
Yeah, when I came, so I started here, I came to WashU a long time ago, July 1st, 1978. I was a first-year resident here at WashU. And at that time, Sam Guze was the head of the Department of Psychiatry. Prior to that, up to about from 1963 to 75, Eli Robbins was the head of the department. But for health reasons, Eli had to step down in 75. So Eli and Sam were still both here in the department, and they ran teaching rounds and things like that. So I interacted with both of them. Now Eli's illness kind of became more progressive, and so he was not as accessible as Sam was. But Sam was a dedicated teacher and met regularly, if not a couple of times a week, with the residents in teaching sessions.
So Sam Guze, and spelled Guze, in case the younger people who might recognize it from his name from a citation. What is there anything in particular when you think about him that you learned from him? Like some 1, 2, 3 things. I kind of remember myself, working with Bob Bick, who very few people had the chance to work with, who was interested in cocaine at the time. And I remember him teaching me that cocaine was not in the DSM-III as an addiction or use disorder, and our research would change that. Cocaine wasn't considered addicting really, probably till the late 80s, but our work did change that. What about Sam and you? What did he teach you?
Yeah, well, Sam was a great mentor, and one of the things, and he wrote a paper that has this in the title of it. One of the things he believed very strongly in is that psychiatrists needed to be tough-minded, which meant thinking carefully about the diagnoses. The diagnosis was the driver of decision-making in the field. It was a statement about the future. The diagnosis was a prediction about what was going to happen to a human being who had certain psychiatric illnesses. And that really stuck with me. So one of their validating criteria for psychiatric illnesses was longitudinal follow-up, that the illnesses should be able to be followed over time, and you should understand the course of that illness so that any treatment that you were going to give to a patient goes against what is the natural history of that disorder. It had a powerful impact on me. And again, this was a radical idea at the time these guys conceptualized it. Radical in a sense, but it was really very conservative if you think about what it is. It's just simply saying this is how all of medicine functions at its best. So that was a big lesson from Sam. A second lesson from Sam, and it actually influenced kind of where I ended up. I ended up as a preclinical neuroscientist, which was a joke, frankly. Mark, I had done zero research before I walked in the door of Washington University in 1978. All of a sudden, they turned me into a cellular and synaptic neuroscientist, But Sam was not a neuroscientist. But Sam, in many meetings with him, it was about the brain was the organ of the mind. And if we were ever going to make progress in psychiatry as a field, we must understand the brain as the driver for psychiatric illnesses. And then I won't belabor all of this, but one other thing Sam did for me was he was the one that got me to go become a preclinical neuroscientist. So when I was a third, fourth year resident, I was unhappy with psychiatry because I thought it was so divorced from the brain that I thought the field's hopeless, in many ways. I mean, I hate to put it in those terms, but that's the way I felt. So my solution to it, being a naive kid from St. Louis, was that I was going to go become a neurologist as well as a psychiatrist. Well, Sam was an incredibly wise and thoughtful and careful individual. He listened to me and met with me several times about this. And at the end of one of the sessions, he says, I've listened to you very carefully. He says, you do not want to be a neurologist. You haven't told me you want to take care of peripheral neuropathies or strokes or things like that. He says, you have an interest in epilepsy, but he says, and maybe that'll sustain you as a neurologist. He says, what you want to do is to understand how the brain works. And that is not what neurologists do. So he was the one that ends up encouraged me and helps me to get into laboratories here at Washington University. So as a fourth year resident, I end up working in a neuroscience lab and end up in the lab of Jerry Fishback, who was a member of the National Academy of Sciences. And it was all goodwill because of Sam. Frankly, when I went to Fishback's lab as a postdoc, the janitors knew more than I was doing. But it was because Sam believed in me that Jerry took me into his lab, and it was life-changing, frankly. So that's the affection I have for Sam.
Yeah, that's just great, and it's great to hear. And it's funny too, just to share with you. You probably know this, but when I was at the University of Florida, I had my second choice was a match in San Diego in behavioral neurology. So I matched at Yale and went there. And my mentor was a former, I think, chairman of neurosurgery at WashU, Al Roten, who's a neurosurgeon. And I thought that I would be a neurosurgeon and was interested in brain systems and drugs of abuse. So it turned out he recommended that I not be a neurosurgeon. And when I met him years later, maybe it was my reunion, 40th reunion or something. And I said, do you remember you telling me that I shouldn't be a neurosurgeon? And he said, of course I remember telling you. I tell that to every person who asks me, but you're the only one that listened.
And psychiatry was better for it.
Yeah, thank you. Yeah, thanks so much. So WashU really pioneered the DSM, and I think while younger people might not recognize that today, it is important, and the research diagnostic criteria move the field along. And I think what you remind us too is that chief residents can actually make big contributions. WashU also, was it a coincidence that it was the center for so much brain imaging science? I mean, I kind of remember WashU being the place that PET imaging was invented.
WashU had, in radiology and somewhat in neurology, had accumulated a number of incredible individuals around that time. Now, I don't know the complete history of this, but Michael Terpagosian was in the Department of Radiology, and he was one of the early pioneers in functional imaging, PET imaging, and things like that. And he attracted some incredible people to work with him. So, for example, Mark Rakel, who was a neurologist actually by original training, ends up in that lab with Terpagosian, and they develop methods for using PET imaging, PET tracers and things. And so WashU puts in the facilities in order to make the PET ligands, which are pretty complex kinds of.
Yeah.
But that was radiology and neurology. And fairly quickly into that mix comes people in the Department of Psychiatry. That Eli Robbins, for example, even though he wasn't as active as he at that time around 1980 because of illness, he had young psychiatrists and scientists around him, and he got them to look at lactate-induced panic attacks as with PET imaging, with 2-deoxyglucose autoradiography, that was the launch of careers like Wayne Drevitz and a whole host of other folks. And Wayne was a little bit later than that, but that was the launching of that. Eli was one of the driving factors behind it.
And just that kind of continued with even current research, connectome and everything else going on. Maybe you just take that up to current times, what the importance of imaging and neuroimaging has been for the department and its research.
Yeah, absolutely, Mark. When we were engaged early, as I said, by the early 80s, when I was a resident, they were doing studies of lactate-induced panic and moved fairly quickly into depression. And that's where Wayne Drevitz and others kind of come on the scene. But the imaging methodology starts to really evolve. It starts with CAT scan, computerized axial tomography, which is kind of a static structural image of the brain, but it moves as you're moving into the late 80s and into the 90s into functional magnetic resonance imaging, where you look at signals that are related to blood flow, and you're able to look at regions of the brain that look like they're interacting with each other. So Mark Rakel and a host of other folks play a big role in the evolution of magnetic resonance imaging. And along the way, they become the drivers of how to analyze systems level kinds of things with network studies and things like that, network mathematics and other things like that to apply to studying that. The challenge with functional magnetic resonance imaging up till very recently has been that while it's really pretty good resolution, it's not good enough in that in order to do studies, you needed X number of patients in the studies. And X, sometimes in those days, the early days, and then into the early 2000s, was only 10, 20, 30 or so patients. Well, it turns out that we now know that the reliability of scans that are relatively short with small numbers of subjects in them is not good enough if you're ever going to get to the level of single patients. So along comes this new generation of imagers that Mark Rakel is still playing a significant role in, but people like Miso Dosenbach, Tim Laumann, who's now in the Department of Psychiatry here, and they develop approaches for what is called precision functional mapping, which is either you've got to use huge numbers of subjects, like hundreds to thousands of subjects, or you've got to do more prolonged scanning of those individuals in order to get reliable signals. Well, that's kind of where we are right now, is the evolution to precision functional mapping and the ability to take the connectome, which evolved along the way, the human brain connectome. David Van Essen played a really key role in that nationally and internationally, among others. But this is all the WashU gang of folks, and I'm not doing justice to the number of incredibly talented individuals who were involved in this. But that evolves through the late 2008, 2010 to the 2010s. Into the present era, and that becomes the basis for kind of how we think about interpreting neuroimaging. And it's really an exciting era for psychiatry, in that we're on the verge of being able to do much smaller number of subjects in a given study, but do it with precision kind of functional imaging. And time is going to tell exactly how this plays out, but that's kind of where the field goes. That's a crude outsider's view of it, by the way. So that's a Zorumski view.
It's a gold standard view. That's fine. Do you ever see imaging confirming a DSM diagnosis?
I hope that that's the case at some point. It may be helpful for predicting certain responses to therapeutics and other things like that, specific regions of the brain. And so while we've been a syndromic oriented department around diagnostic criteria, now we start to think about how does a symptom of lack of pleasure, anhedonia, how does that map within the nervous system? And here I think some of the precision imaging can really start to help. The couple of other trends that have happened. By the way, and I'm curious to get your take on this, Mark, as well. So what I just described to you is that the methods for imaging, fMRI imaging, were good, but they weren't good enough. And so there was unreliability in that. And you need big studies with big numbers of subjects. By the way, that's exactly what psychiatric genetics looks like. Psychiatric genetics is about many genes of low effect size so that you need big sample sizes to get them. So in a way, there's parallel between psychiatric genetics, common disease genetics, and then what's going on with the neuroimaging. But, with modern technology, including the use of artificial intelligence, the opportunity to take EEG signals, for example, as an example, and turn them into meaningful biomarkers is not impossible to see that.
I agree.
Going
I
forward.
agree.
Yeah.
And that's people have trivialized EEG, but all the brain-computer interface work shows that this should be possible.
Absolutely. And psychiatry will be a beneficiary of this, I believe. Yeah.
So, we basically, WashU and your colleagues were really the first to argue and then spend their careers proving that psychiatric illnesses were medical illnesses and that they reflected brain dysfunction and biochemistry. They had an interest in imaging to support this. And I'll get back to this maybe, but the Yale model that I trained, George Henninger and Agajanian, was mostly around the diagnostic system doesn't matter because it doesn't produce a prediction of what you should treat a person with. And Henninger's model was the only diagnosis that mattered at my time in training in 75 was lithium responder. If you could diagnose that, you could save somebody from psychiatric hospitalization and so forth. But let's get back to WashU. So you had the diagnosis, then you had biological foundations of treatment, then you have imaging, but you also have a really major role supporting that kind of idea that psychiatric illness is related and caused by brain dysfunction and biochemistry by the adoption of ECT. So in many ways, and I'm trying to remember this, but I think when I toured the department, inpatient facilities, this would have been in 70. It seemed to me that you had 100 inpatient beds and that most of the patients got ECT.
And I would say that was really correct at that time.
That's funny.
On a couple of things. And so, Mark, the fact that it was about 100 to 110 beds. Now, I can't say about 1970. That's I was way too young for that, right? But by 78, we had 110 beds. That was the size of the inpatient unit at Barnes and a few more. We had another set of beds at the old Jewish hospital, which was just on North Campus. So and in part because of the limits of the psychopharmacology and the limits, frankly, of a number of the therapies that were available kind of at that time. The evidence-based psychotherapy was not a thing that was, in vogue kind of in that area. But the medications were beneficial. Some of the antidepressants, lithium was a beneficial agent. The antipsychotics could be beneficial. So there was the use of all of those. But the gold standard for the department, it still rings true to the present, is that electroconvulsive therapy for difficult to treat, refractory, and complex illnesses was probably one of the wisest things you could do, and kind of move from there. So it was commonly used here. I mean, I as you know, I did a bunch of it during various stages of my career. Matter of fact, if anesthesia would still let us do Saturday treatments, I'd probably still be doing it.
Well, I mean, I'd say that was a position that was evidence-based. So the leaders at WashU who did ECT kept saying, well, compared to what? What is better than ECT? And prove it. And nobody's been able to prove it today, up to today.
Yeah. Yeah, I think that's really correct. Now we have better treatments than we've ever had, but they're still limited, right? I mean, that's the holy grail is how do we break out of where we are and get better treatments on board for patients. Other things that have kind of happened as well. So these days, the idea that you focus on criteria-based categorical diagnoses is really kind of challenged, and maybe rightfully so, in that for Sam Guze and Eli Robbins and that gang of folks, they believed the illnesses would run true over time. So if you had schizophrenia today, you did not end up with another illness at age 50 or 45. And that holds relatively true, but now the comorbidities among the illnesses and Mark, your specialty in substance use disorders, I mean, really speaks to the comorbidities of the illnesses and becomes a real difficult, challenging factor in how you manage patients. And so this comorbid illnesses, including comorbidity of anxiety and depression, which was not appreciated quite as much back in a bygone era, really become kind of the hallmark of the day. So our mindset about therapeutics also has evolved with that.
So I like to look at ECT, and so maybe that's how you developed this clinical research interest in treatment-resistant depression. Is that it? Because that was a clinical interest of yours, and you could then work with Conway and others on vagal nerve stimulation or ketamine or neuropeptides. Is like, where does that all come about?
Yeah, well, it has to do with who comes to Barnes Hospital for care, right? So at Barnes Hospital in its old day and now Barnes Jewish Hospital, we don't see folks who walk in off the street with typical depression. We see everybody is complicated, and they're complicated medically, they're complicated psychiatrically, just as we were talking a few minutes ago. So that becomes the natural, and they usually have failed other treatments, or they're non-compliant with the other treatments because of the illnesses that they have. So we walk into a world of refractoriness, and when you come here to train, that's your world, frankly. And so you learn to think quickly about what are alternative strategies to kind of help. And so a number of folks in the department have picked that up. You mentioned Chuck Conway as one current example. Eric Lenz in the geriatric population, but also even more broadly than that, are all very interested in how do you treat refractory patients and doing high-level clinical trials around it, whether it's Conway's studies of vagus nerve stimulation, Eric's studies of antipsychotic augmentation with aripiprazole and things like that. So this is kind of just a natural for us to kind of follow on. And ECT is still a standard for patients with refractory illness of any type, psychiatric illness of almost any type. So it's a commonly used thing. It remains a commonly used thing here.
And you also have TMS, of course. So when we think about ketamine work, Chuck Conway led an intravenous ketamine and TRD study, and you guys had a prominent American Journal of Psychiatry piece on where ketamine fits in. What do you think about where ketamine fits in?
Yeah, it's definitely not a first line treatment. Because it's got its own complications, and not the least of which is intravenous infusion and side effects of ketamine. So it probably fits in as a second or third line treatment, but probably second line. I think it's moved up on the pecking order, particularly with the advent of S-ketamine. And
yes, Spravato.
Spravato, and you don't have to do the intravenous infusions in that, but yet these things are all fairly complicated to use in some ways. I mean, you've got to monitor the patients much more carefully for a more extended kind of period of time. Certainly with ketamine, you do. So that prompted Conway and the group to take something that John Olney's lab and my lab discovered in the late 1990s, which was that nitrous oxide, so-called laughing gas, is actually an NMDA receptor antagonist, N-methyl deaspartate receptor antagonist. And by early 2010, 2012, Conway and a anesthesiologist here, Pico Nageli, started the first trial of nitrous oxide for treatment-resistant depression. And that's now proceeded through a few clinical trials and in neuroimaging studies that are currently being done. And it's too early to know how well nitrous is going to work. And there haven't been big trials. There have been like 7 trials worldwide kind of the drug that have been published at this point. But it's still too early and too small samples to know where that's going to fit. But these kind of treatments like ketamine, potentially nitrous oxide, other treatments like this give hope that you're now dealing with different mechanisms of action. We're not just doing the same thing over and over again.
I agree. So like nitrous, I mean, you get nitrous at a dentist's office. It's not even regulated by the DEA. Why wouldn't it be a first line treatment to tell if you have basically glutamate signaling or some depression subtype?
Yeah, it's a great question, Mark, and one can argue that some of the things we consider second line. I mean, this is the department, right? When you said how often did they give ECT? ECT was a first line treatment,
right? Exactly.
Walked in because you took the most effective treatment you knew you had and you gave it to patients. So I can see a world where these things all with a pecking order of what treatments you choose will change kind of as time goes on.
Yeah, no, if you had value-based psychiatry or outcomes-based, if you had a different model, like our current model, the patient auditions for the most successful treatments by failing.
Yeah.
It's reversed. And so they get discouraged, they get partial responses, they give up. And maybe we should reverse that. I mean, how dangerous is it to have a test? I mean, what percent of the people have a response on the first exposure to either ketamine or nitrous?
No, it's a good question. I mean, the patients who get it have failed multiple treatments. So we don't really know naive, untreated individuals. You raise a great question, and we can take this from depression and talk about psychotic illnesses. I like the story more in Britain than it is here, that maybe you start for schizophrenia, you start thinking about the use of clozapine very early on. And I mean, clozapine's the atypical antipsychotic. That's the agent that does stuff that others don't do. And so why don't we give that much earlier in the course of illness? So your points about this, Mark, I think are right on the money, which I would kind of expect from you anyway. But
oh, come on. It's just some people think out of the box, and some people don't even know there is a box.
That's a great description.
But I do think about that. I mean, how unique is it to be able to subtype depressions on the basis of some safe test? Anyway, and both of them would be easy to consider. Probably nitrous would be the easiest to consider because it's not controlled.
Yeah, and it also is gone. As soon as you turn off the inhalation, it's gone in 15 minutes, right? Like it is in the dentist office. And so
yeah. I mean, well, anyway, so you guys have done great work.
No, it's a great question though, and a great conversation. And I don't view them as biomarkers for the illnesses. So the lithium story is an interesting one to think about that in that context. But really, since we don't really know the pathobiology of any of these illnesses, frankly, they're just markers of whether you're likely to have a better response to treatment X than treatment Y and things like that.
Yeah, but think about it for a second. You couldn't do it with an antidepressant, even if you had a supposedly serotonergic antidepressant. In the old days, it would take 6 weeks to know if it worked. Then you change the dose. Then the patient would have a partial response. You had no way to even add a biological response into an algorithm.
That's a really good point. And here with these rapid acting agents, you're
Right.
talking 2 to 24 hours. You kind of know whether they're going to have a response to it.
Yeah. So it's just it was on my list of things I could discuss with you. Let's go to like the vagal nerve stimulation. Everybody's interested in the vagal nerve. In fact, we had done some gastric stimulation studies in obesity. There's just what the heck is going on here with vagal nerve stimulation in depression, but just in general.
Yeah, well, Conway has just finished the unipolar study. It's called Recover, and it's the biggest multi-site trial that's ever been done of vagus nerve stimulation. And vagus nerve stimulation is an interesting challenge, right? I mean, it's not a treatment that's going to work right away in
Right.
most patients. I mean, there are probably some that respond quicker, but if you hang in there and the patient will stay with you for going under the treatment with vagus nerve, by several months into the study, you start to see the improvement, and it increases as time goes on to where a year out, you're seeing more responders than you did kind of at even 3 months into the study. Now, Conway's got real data on all of this. And one of the interesting things, and I've seen this with some of Conway's patients, is that those who get better will likely hold the improvement for an extended period of time, meaning some cases years with it, with changes of battery and other things like that. So the treatment, when it kicks in, for the individuals who do respond to it, it can be a longer lived kind of thing. And again, the folks who end up in the vagus nerve stimulation, because it's an invasive procedure, are individuals who failed many treatments.
Yeah, so they might have failed 10 treatments even.
Oh, yeah. That was the original nitrous trial, by the way. I mean, Chuck picked the patients who were the most refractory and put them in the initial nitrous.
Yeah, I always tell them, like, what about starting with something easy? I mean, it's the same thing in TMS. The idea that you had to prove that TMS worked on treatment non-responders better than the second antidepressant kind of set us 10 years back.
Yeah. And now we have, different approaches to TMS, like the Stanford accelerated kind of protocol. This is pretty exciting. Now that protocol is difficult to use, but out of this will come improvements in the methodology. And this is why I think there's a lot of reasons for optimism and excitement about some of the things that can happen.
I mean, I agree completely, and I think maybe there'll be an EEG role here as well. Let me shift gears for a second, because WashU really did the psilocybin imaging pioneering study that I can't remember, but I think it was in Science, and then it was the front page of the New York Times about what psilocybin might be doing that allows it to have some therapeutic effects. And even though I don't believe you were one of the authors, you were the mentor or one of the mentors. What do you think about that work, and where do you think the psychedelics are going to fit? And just bring us up to date onto what Ginger and your colleagues are doing.
Yeah, so that trial was, and this is the precision functional mapping trial. This was a clinical trial based. It wasn't a clinical trial. It was in normal controls, but the clinical trial will be based on something along this line, is what they're moving toward. So this was, I think, 7 patients were and 7 individuals were in the study, and they got this extensive imaging so that you could map their personal connectome. And then they were given psilocybin, and they looked at changes in brain connectivity. And there was a massive changes in brain connectivity, kind of disconnectedness and other things like that occurred, that much of which kind of reverted, relatively shorter period of time, but some changes actually persisted for weeks or so out as long as they followed the individuals out in that particular report. That paper was in Nature last summer, and so it had a big impact because it says a couple of things. One is it's the clearest demonstration that there are these massive brain changes that occur with psilocybin, and that you can look at that in single patients, which is a big step in the future. That's actually where people like Josh Siegel, Tim Lauman, and others are really trying, and Ginger Nickel are really trying to take that imaging approach, is to do this in smaller numbers of subjects because you've got more data and more reliable data for what the connectome of that individual looks like. So that paper has a lot of implications for the future of how we even think about clinical trials in individuals.
Well, that's really important, and part of the reason I wanted to cover diagnosis and imaging, just so we could get back to that with nitrous and psilocybin. It is really interesting career that you've had as chairman and as a principal investigator. I didn't mention any of your neuropeptide work, but I do think you should spend a second at least talking about how that's gone from bench to bedside to approved pharmaceutical.
Yeah. And the molecules are neurosteroids. The ones we work on is, as you know, are derivatives of progesterone that are made in the brain, and they're actually stress modulators. We didn't really know this when we started out. Yeah, right. The way this started out was I partnered with a chemist, Doug Covey, who was a medicinal chemist who turned out to be an expert in steroid chemistry. And so when the first paper, and it actually was paper that the first paper we paid really close attention to was a Steve Paul paper in Science in about 1987. And that paper caught both of our attention because we were working on GABA receptors, the inhibitory receptors, major inhibitory receptors in brain. And on the basis of that, Doug started making molecules, and we were testing those molecules while we were looking for mechanisms of the neurosteroids. By the way, what we thought we could make at the time was an anesthetic. I mean, because many of the anesthetics are GABAergic, so we thought we could make a better propofol is what we thought.
That's funny.
So, and then along the way, it becomes clear that it's a stress modulator. So when the brain is under stress, and I don't mean just psychosocial stress. I'm talking about lower glucose. I'm talking about oxygen changes. I'm talking about inflammatory changes. The production of these neurosteroids gets revved up, allopregnanolone in particular, gets revved up, and it helps to save the pyramidal neurons, the excitatory neurons. So that's a fascinating thing to me. So the neurons that release glutamate, the excitatory transmitter in your brain, make allopregnanolone in order to save themselves from stress. I mean, what a great evolutionary strategy, in my opinion. We fast forward through that, and we're studying the mechanisms of these things with a gang of us here. This was a gang of multiple investigators across 4 departments in the medical center that were all driven by the neurosteroid project. Well, along comes a company that Steve Paul was involved in founding called Sage Therapeutics. And their reason for existence was to see if they could turn a neurosteroid into a therapeutic of some sort. Either for seizures or for, probably not anesthesia they weren't going to go after, but maybe psychiatric indications. And long story short, that company starts around 2011, 2012. By 2019, the drug called brexanolone, which is an intravenous formulation of the natural neurosteroid allopregnanolone, is approved by the FDA for treatment of women with postpartum depression. And then an oral drug follows on the heels of that, a drug that's called zuranolone. And then another neurosteroid has actually been approved by a company called Marinus Pharmaceuticals on the West Coast. It's a drug called ganaxolone, and that's being used to treat pediatric forms of complex epilepsy at this point. So there's 3 neurosteroids that have now been approved, and it's kind of it's kind of an amazing story, actually, because the chances of something like this actually working out are not very high, right? I mean, everywhere along the way, stuff fails and doesn't, new molecular entities don't make it, and so, but here's 3 drugs that have been approved at this point, and that's kind of fun to see.
Well, you see, like from the gold standard point of view, it would not be a surprise because look at what you've done. The nitrous story is a whole career for somebody.
Yeah. And your point earlier about things going both directions. So both the neurosteroid story ends up in humans and it's back into preclinical research.
Right.
Now we're sorting out novel mechanisms for these things on mitochondria and on inflammation and things of that nature. Nitrous oxide, the same way. So, we played with nitrous oxide for about 6-7 years back in the early 2000s, after we discovered its actions on NMDA. Well, Conway and Nageli turned it into potentially a clinical treatment. Definitely. And all of a sudden now we're back in the lab studying nitrous oxide again, so it's this circle of life kind of thing.
No, I mean, it's just remarkable. It's remarkable that so many things have happened at WashU. It's remarkable that so many things happened while you were chair and you were able to continue as a principal investigator. How long were you chair?
Over 25 years, actually 25.33 years, 25 and a third years to be precise.
You know a lot of chairs. I mean, I lasted, I think, maybe 8 years. That was about maybe in the last, but does anybody have your longevity as chair in a major program?
I would bet nationally there are folks who do. I was the longest standing department head of psychiatry here at WashU, but I grew up in an era where Alex Evers, one of the people who does neurosteroid research with us, Alex is a protein chemist, a photoaffinity labeling chemist, but he ran the Department of Anesthesiology for like 28 to 29 years. So I grew up in this environment where, you took the job and if you could make it work and, kept the ship running, you were going to do it for an extended period.
Well, it's just amazing service. Your whole career is just service to mankind. I've described you as the only papal chair. I just find you to be selfless, charismatic, but modest, and the most productive translational psychiatrist that most anyone knows. So congratulations on that. And I'm glad you're still working.
Wow, Mark, thanks. I need you to do PR for me, man.
Oh, well, anytime. You don't want PR.
I'll take you home to my wife. And
that's the level of PR you want. You don't want any PR. I can't even remember the last time you were interviewed. But everyone else gets interviewed.
Yeah. Yeah, I usually say dumb things. So when you guys edit this, you'll figure that out.
They don't have to edit it. It's all very natural. I think since this is a psychiatry audience, it's pretty clear the contributions and the modesty and just the continued success. We wish you continued health and success, and thanks for joining.
Thanks. And walking into WashU was the luckiest thing that ever happened to me. As you know, my wife and I are both native St. Louisans, and she and I knew if I came to WashU, we were never leaving St. Louis. Well, here I am, man, almost 50 years later at WashU. So.
Well, it's been a great marriage and also a partnership with both of you.
Terrific, Mark. And then interacting with you guys all over the years has been a real.
Well, it's a dream for me too. Don't worry. But thanks very much.
All right, Mark.